Okay, terrific. Francesca, thank you very much. Also, Ishir and Felix, who I guess are the three of you are the organizers for this series. It's a wonderful thing that you're doing, and I've tuned into a couple of them in the past. I hope to continue to do that, and so thank you. And I'm looking forward to your questions afterwards. I'm particularly excited to do this talk because it comes at a very good time for one of the two major projects in my lab, which is trying to understand what's so special about synaptic vesicle release, it being so very fast. And this is been the focus of a lot of effort in a lot of labs, and certainly my lab over the last 10 years. And things are just really starting to come together at a structural and a biochemical level, and we're developing some very precise ideas and hypotheses that I'd like to share with you today. Francesca, could you give me a thumbs up if you can hear me okay? You can hear me?
Everything's good. We see and we hear perfectly fine.
Okay, I just want to make sure.
Yeah.
Okay.
Yeah. We'll stop you if there is something wrong. Yeah.
Okay, thank you. So, yeah, I think as all of us know, it's a little hard to give a lecture or talk on Zoom because you don't get, the kind of feedback you normally do from a group. Okay. In any case, I think we all understand pretty well that the fusion machinery consisting of snare complexes or snare pins exerts force on two membranes, pulls them together, and results in fusion. And that's a physical process that is used over and over again in nature. And it underlies, I'm hoping you can see my pointer here. It underlies, as you know, vesicle trafficking on the exacidic and endocytic pathways. It underlies the more controlled, temporally controlled release of regulated secretions in the exocrine and endocrine systems. Okay, someone else would like to do a recording. That's fine. And it also underlies the most specialized version of triggered release, which is in fact the release of the synaptic vesicle at every nerve ending. And I think you'll remember that the synaptic vesicles contain the neurotransmitters. They accumulate on the presynaptic side of a synapse, separated from the presynaptic and the postsynaptic membranes. Here shown is a neuromuscular junction. That's a muscle cell, but it could equally well be one nerve synapsing on another. Each synaptic vesicle contains about 10,000 molecules of neurotransmitter at a concentration approaching 100 millimolars, so it's a very high concentration. And the resting calcium in the nearby cytoplasm is perhaps in the range of a few nanomolar. But when an electrical signal comes down the wire, so to speak, the axon, it causes the neurotransmitters to be released, and to do so very, very quickly, which is the point that we want to understand. How can it be so very, very quick? And what in fact happens, in a little bit more detail as background, is that nearby voltage-gated calcium channels, literally within molecular contact distance frequently of these vesicles, open up in response to the arrival of the electrical impulse. They emit calcium to the nearby cytoplasm, which rises rapidly within a millisecond, certainly to about 100 micromolar in that general range, and then is rapidly sequestered by local mitochondria, endoplasmic reticulum, and pumping out of the cell. And so you get a wave of calcium that rises and then falls equally as rapidly. Synchronous neurotransmitter release results when these vesicles that are pre-bound and ready to release, they're called readily released pool or ready release vesicles. When they experience this rapid calcium rise, the calcium sensor protein, which is in the vesicle, the synaptic vesicle itself, is called synaptotagmin. Synaptotagmin complements the snares by adding calcium regulation in a way that I will describe. And the wonder of it all is the sheer speed. And that's the main point of the slide, which is that literally the same snare proteins, or virtually the same snare complexes in physical terms, energetic terms. The snare complex by itself takes about 1 second to fuse two vesicles. Which is more than adequate for most transport pathways like Golgi or endocytosis. It's certainly adequate for the release of insulin and most hormones, but it's hopelessly inadequate for the release of neurotransmitter. If our synapses were this slow, we would be even stupider than we are. In fact, the vesicles are released starting as fast as, and maybe even faster than 100 microseconds. So there's a 3 or even 4 order of magnitude disparity in the speed here. And that has long been a problem for neurophysiologists who say, well, your snare proteins, they probably are important, okay? And we know they're important for the synaptic vesicle release, but are they really enough to explain this extreme rapidity of synaptic vesicles? And so I think my answer today will be, the answer is basically yes, but as a result of a supramolecular organization in which, in fact, exactly 12 snare complexes, and I say that number based on recent data, are cooperating spatially and temporally to achieve cooperativity in release of the neurotransmitter. And as a result, they can act synergistically in both a kinetic and energetic fashion, okay? So that's the high-level message, which is that you can explain all of this on the basis of snare complexes, but the explanation has taken on a life in and of itself, and has introduced a whole variety of new types of regulation that we can think about that are probably unique to the neuronal synapse. So I'm now going to move on with a bit more detail. Now, this journey down this path began about almost 5 years ago, when, in collaboration with my Yale colleague Jean-Lu, we made an unexpected discovery. I don't think anybody would have expected that underneath every one of these vesicles that you saw a moment ago, and you've probably seen for decades, that underneath every one of these readily released vesicles, there might be a structure. But there is. There's a structure. We only catch a glimpse of it here because currently the resolution of cryo-EM tomography in vivo in this setting is still relatively low. But it's a high enough resolution to see the symmetry of the structure. So this is a side view of a synaptic vesicle. It's an average, of course, of thousands of synaptic vesicles, an unbiased average. And you see that there are densities here that actually are found at a spacing going across the vesicle in cross-section of about 30 or so nanometers. 40 or so nanometers. And if you look at the top view, so that would be like cutting a line right here and looking from the top, you see that these densities are organized on a ring. You see actually that there are exactly 6 of them, and there are 60 degrees between each one. So we have a 6-fold symmetrical structure that implies that the snare proteins, along with all the other fusion machinery that's there, the molecular chaperones and so on, and the calcium sensor, synaptotagmin, must somehow be organized in a ring-like symmetrical structure that extends over the full size of this object, which is 40 or 50 nanometers in diameter, okay? That's an extraordinary conclusion. So what might be the structure of this supramolecular complex that evidently exists in and around or between the two membranes of the synaptic vesicle and the plasma membrane? What could possibly be its structure? Well, I think we're all aware of the recent work on nuclear pore structure, where X-ray crystallography has been put together with cryo-EM tomography outline, okay, of the overall shape of a structure, and with enough information to allow the proteins, the X-ray crystal structures, to be plugged in. This cryo-EM picture is not of that level of resolution, but it does provide enough initial information to start orienting the crystal structures, even though it is very low resolution. And the main clue comes from the dimension of the object and from its six-fold symmetry. So let's take an unbiased look at what the X-ray crystallographers have produced over the last 20 years of the structures of the known genetically and biochemically validated working parts that we know to be present at the same region from biochemistry and from immunoelectron microscopy, okay? And let's start with the calcium sensor, synaptotagmin, and of course, the well-known four-helix bundle of the snare complex, consisting of a V-snare, a helix that comes from the synaptic vesicle, the molecule VAMP2 or synaptobrevin, the syntaxin molecule, one of the T-snare subunits, also an integral membrane protein attached to the plasma membrane with a TMD, which is not shown here. This is just the cytoplasmic portion. Okay, and then SNAP25, shown in green, which contributes two helices to this four-helix bundle. We also have the working end of the synaptotagmin molecule. This is a transmembrane domain protein, which is embedded in the synaptic vesicle. And in this structure from Axel Brunger and Tom Sudoff, we see the way in which one of the two C2 domains of synaptotagmin in the cytoplasm binds in a very specific fashion to the surface of the snare complex. In fact, it binds to a half-zippered snare complex, which we call a snare pin. Others call a trans-snare complex, which is engaged in the process of fusion. And we know from genetic and physiologic studies, as well as biochemical reconstitution studies, that this binding reaction is necessary to freeze the frame of the zippering to prevent the snare complex from zippering completely. In other words, this is what we call clamping. This is the interaction is needed to clamp in some fashion, we don't know exactly how, to prevent the further zippering of the snare, to prevent the fusion from occurring. So that when calcium binds to the calcium binding site here, that can cause this synaptotagmin domain to fall off and now release the snare complex to complete the fusion. So that is kind of a certain level of understanding of this structure. So this structure is very important to include. There's another very important structure, if I can say so, because it was our structure, from my lab and from especially Karen Reinisch, my colleague at Yale, from her lab. And we published this back in, I think, 2011 or 2012. And it became controversial from the very beginning. And it's only in the fullness of time over the last 10 years that the genetic and biochemical evidence validating the structure has accumulated, okay, in a singular and a forceful way. I won't have the time, obviously, to go through that, but you have all the references here if you need them. And the important part of this complex is what it demonstrates is a pairing of two snare complexes asymmetrically in the crystal. Here's one snare complex, here's another, and you'll see that these two snare complexes are actually bridged. And they're bridged by another molecule called complexin. This molecule is needed both to activate the assembly of the readily released vesicle, and it's also needed genetically to inhibit or clamp the further progress of the snare towards fusion as it's hanging around waiting for calcium. Now, mutations, for example, in this accessory helix portion cause the vesicle to become unclamped, and spontaneous fusion goes up in a variety of different animals, as well as in vitro systems. The same result happens when you take away synaptotagmin in a similar setting. So both synaptotagmin and the complexin molecule through its accessory helix contribute to the clamping of the snare complex. The two snare complexes that are recruited together or paired are held together because the central helix, the adjoining portion of complexin, binds to one snare pin, while the accessory helix binds to the other. Now, I want you to zero in for a moment on this interaction right here. I'm hoping you can see my arrow. What this blue here is a V snare that is half zippered. The reason it's only half zippered in the crystal structure is that we did not include the rest of the V snare in the synthetic peptide. So as a result of that, the accessory helix can easily bind, and it doesn't have to compete. Normally, that V snare would be coming out here, and it would be competing with the accessory helix for binding. And I'd like you to see that here, the accessory helix is bound as in the same place that the V snare would be bound if it were in the 4-helix bundle. It actually occupies the same space in the pocket defined by 1, 2, 3 helices locally of the membrane proximal T snare. So in other words, the complexin helix is like the finger in the dike. It is plugging up the hole where the V snare wants to zipper. That's why it clamps. And when you remove it, now the V snare, when it's present, can complete the zippering. And that will happen if this snare complex with the complexin attached to it here were to move away. Okay? And you'll see in a moment what the relevance of all this is. So this is a second structure. Okay? And the third structure we discovered again around 2012, 13, and we've developed over the last decade quite a considerable body of genetic and biochemical evidence for its physiologic relevance, and it is a ring of synaptotagmin. In fact, this domain right here, the C2b domain that does the calcium sensing, that domain by itself will self-assemble in the absence of calcium to form a ring. And guess what? The ring, although it's variable in vitro because you can have different numbers of copies, from 15 to 25 copies of C2b, the ring has a diameter in the same range, 30 to 40 nanometers, as the ring that we discovered later to exist under the synaptic vesicle. So you can see it's a very appealing idea that maybe we could assemble the snare complex here, not just one, but as a pair, onto this ring, and maybe that would begin to explain the underlying structure. I also want to point out before I forget that while this ring assembles in the absence of calcium, it disassembles in the presence of calcium. So it's a calcium-sensitive ring, which introduces some very interesting ideas. Now, how can we snap these structures together? Well, I think I've already indicated how that could work. We'll start with the asymmetrical pair of snare pins and the synaptotagmin ring. Well, all we have to do is actually take the bridging structure then to try to pull those together is this, because it tells us how to bind a snare pin to a synaptotagmin, which can then be in the ring. Now, of course, these structures don't have to be compatible, but actually they are. So you literally can take the coordinates from the Brunger structure, attach the C2b domain right here, following that prescription, literally load one of those subunits into here without affecting the bonding to the neighboring subunits of the ring of synaptotagmin, and you produce this structure. More satisfyingly still, if you would take the C2b attached to the transclamp snare pin, unlike this structure, it no longer fits in to the ring. So there's actually only one way to snap this together, and that is with C2b bound to the snare pin, which has the central helix bound to it, okay? This one. This helix, this snare pin here, which is structurally different than this is the clamped snare pin. This has the central helix on it. I'll call it the central snare pin. And that central snare pin is the only one that with synaptotagmin, according to this recipe, can drop in here. If you try to force synaptotagmin over here, it cannot be forced in there sterically. So there's only one way to snap the structure together, and that's this. And it's a very interesting result. Looking from the top, you see that the central snare pin because it has the central, named because it has the central helix of complexin on it, is central in a second way. It literally is on the calcium sensing ring, and its transmembrane domains, which would be going up towards you, looking at the screen into the vesicle, or down into the plasma membrane, they are both located within the ring. The other snare pin, which is transclamped by the accessory helix of complexin, is now radially outwards. I hope that's clear to everybody. We could therefore call that the peripheral snare pin. Central in this hypothesis, because this is, of course, speculative, peripheral, okay? And the idea is now, very simple, that the central snare pins, whatever number there are bound to this ring. As long as they're bound to this ring, they can't zipper any further, because if they zipper up, they start moving towards the contact point where fusion will occur, and they can't do that. But when calcium comes in, the ring will perhaps cooperatively disassemble, and as it disassembles, these central snare pins can now continue zippering. And so that's the model of how calcium regulation occurs. When the central snare pin continues to zipper, it will remove at the same moment the accessory helix from the peripheral snare pin, which will now also be able to zipper. So in this model, the central snare pin is directly regulated by calcium because it's bound to the calcium sensor. The peripheral snare pin is indirectly but contemporaneously regulated by calcium because it is mechanically coupled in this Rube Goldberg machine through the accessory helix, okay? Now here's a view from the side, and you can see that, again, very satisfyingly, the geometry places this entire structure. It goes off of the plasma membrane, and it seems to follow the synaptic vesicle rather closely on the outside. It doesn't bump into it sterically. That's very important. And here you see it from an oblique angle, which makes clear how the accessory helix does the clamping of the peripheral snare pin, okay? And you can see that the peripheral snare pin actually, if you were to look in the detail, likely has its transmembrane domains outside of the synaptotagmin ring. That's a point that we cannot say with certainty, however. Now, left unclear from this is this, so the idea would be that there's some number of these. So the basic idea that emerges is that the snare complexes or the clamped snare pins in the readily released vesicle are already arranged in pairs of peripheral and central, and they are molecularly paired together by complexin, to repeat. Now, the other main fact that came out of the cryo-EM is that there is six-fold symmetry. So that would imply that somehow or other there are 6 of these structures that are arranged on this ring, okay? Not 8, not 12, not 4, but 6, okay? That's a strong prediction. Is there any other protein? Because there are many other proteins. This is just some of the other proteins. To list the other proteins, you know that syntaxin is regulated by MUNC18 that keeps it in a closed state. You know that there's a key chaperone called MUNC13, which is known to react with the closed state of syntaxin to remove the MUNC18 locally and open it up so it can react with the other snares. And we now know also that MUNC13 binds all 3 snares. It binds VAMP, syntaxin, and SNAP25, and it is needed in a concerted reaction demonstrated a few years ago by Jose Rizzo first, but validated in many labs, that allows all 3 snares to come together at the same moment. Details unclear, but that much is clear. So we need to at least add to this picture, and I will, MUNC13, MUNC18, and there's one other protein which is very interesting from the point of view of symmetry, and that is shown here. It's called synaptophysin. Now, synaptophysin you may or may not have heard of because you don't hear too much about it, okay? It's kind of like the black sheep or whatever of the family. It's one of the most abundant proteins in the synaptic vesicle. It's actually used by developmental biologists and cell biologists as a marker, a marker in immunofluorescence and so on for the synaptic vesicle. It's that unique to the synaptic vesicle There are 30 copies of it in every synaptic vesicle. There are, for comparison, 70 copies of the V-snare vamp in approximately in each synaptic vesicle, according to the classic accounting of Reinhard Jahn in his lab. But this is not a typical synaptic vesicle protein. It's a 4 transmembrane domain protein. It's also untypical in that we know so much about the other synaptic vesicle proteins, but we actually don't know what this protein does. Isn't that sort of amazing? And so why don't we know that? There's 2 reasons. One is that there are no mutants. And 2, as a result, nobody studies it. There's no biochemistry, practically. Okay? Now, if you knock it out of a mouse, the mouse is fine. That, of course, means it's of no interest to geneticists. And since genetics dominates the world today, it's been assumed that this protein is of no interest whatsoever. But that may not be true, because in the last few years, several laboratories have undertaken to study what happens in the knockouts. And it turns out there's a whole series of molecules closely related, synaptogyrins and others, and they start popping up. And we're just beginning to fathom what goes on in those knockouts. So we don't really know. But we know enough to know that actually the negative result that has caused this field to be stagnant for 20 years may be overstating the case. It's also turning out, and much of this work is not yet published, that there are human disease, rare human disease triads or trios that result through spontaneous mutations in synaptophysin, which is an X-linked gene, and therefore more prone to the effects of spontaneous mutation. And these are now being tracked, and there are very clear synaptophysin phenotypes in the central nervous system in the rare, unfortunate person that has a mutation. So it's very clear now that synaptophysin is an important protein. What could be its function? Okay, well, the hero in this field is named Stowell. He's at the University of Colorado in Boulder. And about 15 years ago, or even 20 years ago, he started studying the biochemistry. And it's really his lab that has done all of the biochemistry here, mostly not recent. And we've followed a bit in his footsteps and validated, using more modern techniques, really, everything that he's concluded. And his first conclusion is that synaptophysin is a hexamer, which is why I'm telling you all about this. It's why I got interested in it. And if you isolate it, it's a hexamer that dissociates. Interestingly, it requires cholesterol to stay as a hexamer. It's sensitive to some detergents, not others. We've used single molecule methods, I won't show you the data today, to validate that it is a hexamer when it's purified and reconstituted in bilayers in the presence of cholesterol under native conditions. And most importantly, we validated that, and this is an example of a single molecule experiment done in detergent solution by Manindra Bera and others in the lab, where we have synaptophysin with one color, it's a fluorescent protein tag, and pure VAMP2, these are all pure proteins mixed together. And we allow the complexes to come down on cover slips, standard methodology. And we ask, how many copies are there? So here you see, for example, in photobleaching, you've got one photobleaching event, one molecule of synaptophysin. This one had 1 copy of VAMP. This one had 2 copies of VAMP. And in general, you get 2 copies of VAMP, sometimes 1, but never more. So as was determined biochemically by Stowell, this molecule of synaptophysin, this protomer of synaptophysin, one subunit of the hexamer, strangely enough, binds 2 molecules of VAMP, which is an asymmetrical binding. You'll see eventually today, hopefully I'll get to the data, that it's actually VAMP is forming a dimer through its transmembrane domains, and one such dimer binds to one synaptophysin. Stowell further found this by electron microscopy that the molecule is not only a hexamer, but a symmetrical hexamer by negative stain. We've extended this to cryo-EM. Here's our cryo-EM low-resolution image here. This is the cytoplasmic domain, which is actually an amyloid-like sequence, which seems to additionally hold together the hexamer. The hexamer falls apart, by the way, effectively in the absence of VAMP. And so you can see that while you can imagine that the hexamer will be in the synaptic vesicle at the contact point, And of course, it needs to come apart at some point during fusion, and so it makes sense that it would be dissociable. So obviously, you're thinking now, as we were thinking, that, well, we know that our structure has six-fold symmetry. There's conveniently a hexamer that may be there in the middle of all this, and it conveniently binds two vamps, which could mean at each vertex, which could mean one pair of snare pins, given everything else that I've proposed. And that's exactly what we suggest, and that's encapsulated here. So now this is a space-filling model that shows the asymmetrical pair of snare pins, peripheral, central, and with the complexin linking it, central helix, accessory helix. And all we've done here is taken the structure that I showed you before, and now we've added in 6 of these, and we've added in the central synaptophysin. So now you're looking from the top, and this is now the vesicle membrane. We're not actually showing the vesicle, but you see the yellowish part right here? That would be the transmembrane domains of synaptophysin. This is a synaptophysin molecule. This part right here is in the lumen of the synaptic vesicle. That's the cytoplasmic domain. So picture the synaptic vesicle membrane going like so. You're now looking down on the plasma membrane. Let's watch it loop again. Synaptic vesicle goes like this. Plasma membrane is below. Here's the synaptotagmin ring on the plasma membrane. Every 60 degrees, you would then have a central snare pin and a radially upward projecting, blanketing the synaptic vesicle, peripheral snare pin. Okay? So I hope this is pretty clear. This is a three-dimensional view, and you see it already has a lot of subunits. In order to make this fit the structure that we have, we actually need 18 rather than 21 or 15 copies of synaptotagmin. I won't have time to go into that. 6 copies of synaptophysin, 12 copies of VAMP, 1 with each paired snare pin, 12 syntaxins, 12 SNAP25s, and each snare pin binds a complexin. Now, although this is complicated enough, it does not include MUNC13, MUNC18, and it does not include the other major domain of synaptotagmin. We don't know where to place the C2A domain. We actually have no clue at the moment. Okay, now, what about MUNC13 and MUNC18? Well, an independent body of work, largely carried out in the lab, structural biology work by Kirill Grushin, a senior research scientist in the lab, and aided biochemically through, and I'll show you some of this data momentarily, by others, most notably Venkat Sundaram and others in the lab, and I'll give them credit as we go along, has characterized previously unknown structures of MUNC13. Now, I've already introduced MUNC13 to you as a molecular chaperone that is critical for assembling the snare pin, involving all 3 snare complexes. And it has the MUNC13 molecule has 4 main domains, and I'm showing you here a space-filling model, and just focus on this one right here. It's 1 copy. These are all identical copies. There's a C1 domain that binds diglyceride. This is the plasma membrane. There's a C2 domain called C2b that binds calcium and PIP2. There is another C2 domain up here called C2c that binds the synaptic vesicle. It binds phosphatidylserine in the synaptic vesicle. So you can already gather that this molecule is known, and it is known well before our work, mostly the work of Jose Rizzo, to span and actually capture the synaptic vesicle on the membrane. And then it's got this very large 20 nanometer long, synaptic vesicle is 35 nanometers, 20 nanometer long, nearly as big as a synaptic vesicle long, MUN domain, M-U-N, consisting of 1, 2, 3, 4 repeats of subdomains. And these subdomains bind the various snare proteins. And what was not known until our relatively recent work is that they also bind each other in previously unknown ways, so that this molecule of MUNC13 or MUN can actually bind itself in different ways to form oligomers. And there are at least 3 oligomers that we know of. 2 are directly demonstrated, and the 3rd is implied and not yet directly demonstrated. 1 oligomer is an upright form, and it's involved in capturing the vesicle. The next oligomer is a hexamer, in which these subunits rearrange, I'll show you that in a moment, to bind each other. And the C2C domain that was binding the vesicle, I think you can see, is now engaged in binding a MUN domain. The vesicle now binds to a different surface of the hexamer. And so what's happening is the vesicle was at 18 nanometers separation. It's now gotten to 12 nanometers separation. And in the next transition, the vesicle comes even closer to the membrane and forms the ready-release vesicle. And what's really going on here is that MUN13 is acting as a scaffolding protein, which rearranges itself, and believe me, I don't think this is the end, it's only the beginning, through a series of oligomeric transitions that structure the assembly of the snare complex. So they actually prevent the assembly of the snare complex in the captured vesicle. You'll see some examples in a moment. I hope to get through this, in which in the first stage, they assemble the 6 peripheral snare pins. Because remember, what I'm really saying here is the MUN13, you can easily get confused here. MUN13 is sort of structuring the overall process. The end game is to produce the structure that clamps the snares to allow for ready release. But in order to get there, the structure, the assembly of the structure that I just showed you, that we have postulated but not really proven yet, okay? But if you keep it in your mind that it might exist, that structure forms in a series of discrete stages that are structured by the scaffolding protein. And therefore, the oligomeric states that you see here, the upright, the lateral hexamer, and now what I'll eventually call the buttress ring, or the flat ring, they allow or don't allow snares to assemble, and they direct the assembly of the snares into the end of synaptotagmin, probably into the structure that finally results, that we saw a glimmer of by cryo-EM. And so there are at least 2 stages that we've now predicted and to a large degree validated. 6 peripheral snare pins assemble at this stage, and we call this readily mobilized. And then the final stage, which I showed you a moment ago from the synaptotagmin and from the snare point of view, readily releasable, where the separation of membranes is less than 5 nanometers, probably 3.5 nanometers, in which the synaptotagmin ring is assembled. It was not assembled here. There are 6 central snare pins, and in addition, the previously assembled 6, so that you have a fully assembled, ready-to-go structure. Stage 1, Stage 2, Stage 3. These transitions are regulated. This transition is regulated by calcium. What we really have is an assembly line in which this readily released vesicle is discharged with calcium. It then needs to be replaced. That same calcium wave triggers the readily mobilized vesicle in this model to replace it. And on a rather different time scale, the same wave of calcium triggers the hydrolysis of PIP2 to release diglyceride. Diglyceride binds to the C1 domain, and in so doing, flips the entire molecule in this direction to cause this transition to take place on a different time scale. And the result is a spatially and temporally regulated pathway that sets up an assembly line, the end game of which is this readily released vesicle. At somewhat higher resolution, and by the way, I haven't told you where this comes from, we have captured in the form of a crystal between 2 bilayers the pure MUN13 protein, and we've captured it in these oligomeric states. We've solved these structures. We've taken the novel interfaces between these. This is all published. We've taken the novel interfaces such as this interface right here. Actually, the molecule, the crystal that we start with, in fact, is not just this molecule of an upright MUN13, but you see it's in fact a trimer. I'm hoping everybody could see that. Let's wait for this to loop again. So the actual oligomer we see in our structure is shown right here. It's an 18-mer in which 6 upright trimers are arranged along a ring. Interestingly, the dimensions of the hexagon right here of trimers is exactly the same as the dimension of this hexamer. and is the same as the width of the cryo-EM tomography structure. So you see the coincidences are starting to stack up. And we don't think they're coincidences. So we think what's happening is and we've actually taken this novel trimer interface that holds together. You see what's happening in the trimer. Let's let it loop again. This trimer is formed when the MUN domains contact each other through this region right here, okay, through that novel interface. We've mutated that interface, and reconstituted synaptic vesicle exocytosis is dead. Okay, I won't be showing you that. In fact, I'm showing you rather little data, as it turns out. And in collaboration with our nematode collaborators, Jeremy and his colleagues have in fact found that nematodes are unable to release neurotransmitter when you mutate this interface or this interface. Okay, so we're very clear on the physiologic relevance of these oligomers. And so you can see that you can transition from the upright to the lateral hexagon simply by a motion which is induced by diglyceride binding to C1 that allows it to rotate. So let me go to the next slide. So I want to just pull all this together, and I will take a couple minutes now to really, like a buzzsaw, go through some data so that you will see that there's some reality to this. This is an excerpt of the readily released vesicle, showing now an inner ring of synaptotagmin, an outer ring of MUNC13, as the MUNC13 lateral hexamer, as the vesicle is brought down to the membrane. An inner ring of synaptophysin, which we think is the organizer for all of this, okay? And now showing the transclamped pairs of snare pins interacting with the clamping ring of synaptotagmin, the calcium-sensitive ring, but quite possibly also interacting with the 6 MUNC13s that are arranged on the outside. Now, as promised, I'm going to quickly go through data that I think you'll now be able to understand very quickly and efficiently. So here we use an in vitro system, which includes all of the known proteins in pure form and monitors the release of, again, it's published if you want to see it, and includes and monitors the release of fluorescent dye across from single vesicles on suspended bilayers observed by TIRF or confocal light microscopy. I'll show you a few more details of that as time goes along. But what I'd like you to just see is we're measuring the number of vesicles per 100 micron square of membrane here in red with all wild type proteins. This is, and we're measuring how many are clamped. And now we're measuring at the same time how many released over a matter of a few minutes. So this measures spontaneous release. Here we have a mutant of synaptotagmin that controls the assembly of the synaptotagmin ring and does so both in vitro and in vivo. Here in vitro, you see spontaneous release goes way up. So this shows that ring assembly of synaptotagmin is in fact necessary for clamping, as we model would predict. We have another recently obtained mutation that obtained by Aturali Chatterjee and Kirill Grushin in the lab, in which MUNC13 assembly is affected. And you can see that assembly, most likely of this MUNC13 ring, is also necessary, equally as necessary, to clamp the readily released state. So the clamp requires both MUNC13 and it requires synaptotagmin ring assembly. And I think I've already explained why that is, because the ring of synaptotagmin, for example, dissociates with calcium. This is all known. And in vitro and in vivo, when this happens, the snares are now released and they can cooperatively complete their zip ring. I think I've also mentioned that when that release occurs, our model predicts that this accessory helix is removed and that the peripheral snare pins are also therefore going to be released. So therefore, if you make a mutation that compromises the accessory helix, and this binding reaction right here, be it in vitro or in vivo, and we've tested both, you should now get spontaneous vesicle release. And this is recent data, again, from This is from Amit Aji and Munindra Bera in the lab. And look what happens with the complex and accessory helix mutation. You'll see in the same in vitro system that spontaneous release goes dramatically upwards. And now I've added another element because AlphaFold has predicted for us that the transmembrane domains of VAMP should actually bind each other to form a dimer. And VAMP has been known for 2 decades in detergent solutions to form a dimer. But AlphaFold has predicted certain mutations that could be made that would affect dimer formation. And we've made those mutations, and they, in fact, disrupt dimer formation. And we've therefore taken those same mutations and placed them in VAMP in this reconstituted system. And you see that when we disrupt dimeric pairing of VAMP, we also disrupt the clamp. So our current thinking is that the pairing of peripheral and central snare pins is not only undertaken and produced by the complexity, by the accessory helix and by the bridging complexin, but also within the synaptic vesicle membrane, they are also paired from the very beginning. And remember now that synaptophysin binds, we know, two copies of VAMP, and we suspect a pair of a pre-paired VAMP dimer. So now you see how all this fits beautifully together. And you see that synaptophysin may also be contributing to the clamp by stabilizing this dimer, which stabilizes this structure and keeps the finger in the dike to prevent it from releasing. You see that we now have very good evidence of that, okay? In vitro. The in vivo tests are now underway. This is meant to illustrate more particularly the reconstitution system in which we have, there's an annoying panel here, let me move it down, in which we have a suspended bilayer, this meaning it has water on both sides. It's formed from a giant unilamellar vesicle. The GUV is laid down on a chip, which has, here's a chip with many holes in it. And in those holes, we have water coming from below in these holes, and these holes are actually filled in with OptiPrep iodinated that raises the refractive index to the point that matches glass. So it looks at, so this, as far as the cover, this bilayer is concerned, it's sitting on glass, but it's actually sitting on water mostly. These holes are actually about a half a micron, unlike what's shown here. We have to get another drawing this to scale. And so what you have is many holes, hundreds of holes on a chip covered by 10 micron, 20 micron GUVs, which cover many holes. In the GUV are the T-snare and the other plasma membrane components. So that's MUNC18 and syntaxin, along with palmitoylated SNAP25. We add diglyceride. We have PIP2 here. The vesicles contain physiologic numbers of copies, physiomimetic copies of outward-facing synaptophysin and reconstituted VAMP, and synaptotagmin. We're adding complexin and MUNC13, that is recombinant pure protein. So you can imagine that this is actually in itself quite an accomplishment to achieve this. It represents the work of Venkat Atruli, and when he was in my laboratory, and now independently, Shyam Krishna Kumar, and they have succeeded in this, actually the only reconstitution that I know of in the that involves a planar membrane, and it's very critical to have a planar membrane. Other laboratories have successfully reconstituted this fusion process, notably Rizzo, Sudhoff, and Brunger. There are probably others, using two vesicles. That's much easier to do, but you can't count molecules that come in two vesicles because the vesicles already contain, at optical resolution, all the molecules there are going to be. But if you're looking down at a bilayer and you look at a docked vesicle, you can see when other molecules come in because they come into the field. And so you can actually measure release here by having either labeled dye, typically sulforhodamine B in the vesicle, typically self-quenching, not so much that you don't see the vesicle appear. Here's a trace of what you see when the vesicle appears, then when the vesicle fuses after you add calcium, it dequenches and then diffuses away. Or you can have phospholipid in the wall of the vesicle. and you see then the vesicle docks, and when the vesicle fuses, the lipid bilayer diffuses away. So that's the measurement. You can measure and count snare complexes very simply by adding fluorescent complexin. When because we're using turf in this instance, the complexin that's up here doesn't show up. It only shows up down here, and only then when it binds. So you can actually count snare pins coming in. So here, for example, is an experiment where we've watched the vesicles that have docked, and after they've docked, we then can add calcium. They get released. I'll show you some examples of that in a minute. But here what we've done is we counted the number of snare pins on the vesicle by counting the number of complexins that have bound to the vesicle, one per snare pin. You'll see that if we have a complete reconstitution, look at the histogram, the frequency with which we get a certain number of snare pins versus the number of snare pins. We have 12, a very tight distribution, 12 plus or minus 1 snare pins. Pretty amazing, actually. Now, if you leave out synaptophysin, it's the only protein you leave out, you get between 5 and 35 snare pins. In this case, we've used pre-assembled snare pins. It simplifies things for various reasons. But you see, simply adding synaptophysin templates 12 snare pins per vesicle. So now we have established the first biochemical role of synaptophysin, which is, as predicted, to control the number of snare pins. And there are 12 snare pins, 6 pairs of peripheral and central snare pins that are templated. Actually, the temporal resolution and the sensitivity of this method has allowed Manindra Bera, who developed the method, to actually count the number of snare pins in kinetic experiments as they come in. So what we're doing here is looking at a single puncta on a bilayer, meaning a single vesicle, the membrane of which is fluorescent. And that's in green. So we can see the fluorescent phospholipid in the tracing. There was nothing. The vesicle now docked and became immobilized. But let's look at the number of snare pins. We first see the near synchronous assembly of a certain number of snare pins on this vesicle, 6 in fact, then a lag phase, and then you'll see a second wave of snare assembly, a second 6. This happens to be now a second example of a separate vesicle. Here you see the vesicle docks, boom, stays docked. By the way, if you add calcium, which we always do to make sure that everything was right, this vesicle will synchronously release and it will fuse. So we know it's a competent vesicle. Now you see here 6 snare pins form, lag phase, typically a few tenths of a second, and then second phase, 6. And if you look at the total, you'll see if you include both waves, it's 12 plus or minus 1 snare pins that they formed in 2 equal waves of 6 plus or minus 1, 6 plus or minus 1. So isn't that interesting? So the obvious explanation is you're forming one set of snare pins first. We think they're peripheral for various reasons, but we can't absolutely prove that today, followed by central. The reason we think the central ones form second is that the synaptotagmin ring must assemble for them to form. How do we know that? Here's a series of experiments. If you leave out here's what you just saw, 2 waves of wow. Yes, okay, 2 waves. Here's what I just showed you, wave number 1, wave number 2. Now, if you leave out synaptotagmin, you just get 1 wave gated by synaptophysin, and it's precisely 12. If you use a synaptotagmin mutant that doesn't bind a snare complex anymore, you now get 12 on the average. You lose precise templating, and you only get 1 wave. If you also only get 1 delayed wave, you don't do away with the delay when you have a synaptotagmin mutant that doesn't form the synaptotagmin ring, but allows synaptotagmin to bind the snare complex. So the conclusions then are synaptotagmin causes the lag phase. You see no lag phase without synaptotagmin. That what's causing it is the binding to synaptotagmin. and that the ring is needed to precisely for synaptophysin to precisely template 12 snare complexes. So for that reason, because of the connection to the ring, we think that the second wave may be the central ring forming. First peripheral, then central. We think that, but we don't know that. Now, the final point I want to come to, and if I can take a couple more minutes, is to introduce another idea very quickly, and that's a physical idea. Why is it that there are peripheral and central snare pins? And I'd like to come to the core question, what turbocharges neurotransmitter release? And I'm going to make the point that the turbocharging, the big difference in speed between the synaptic vesicle and everything else, is going to be due to these peripheral snare pins, not the central snare pins. Essentially, what I'm going to say is every fusion process is going to end up having, probably, we don't really know that, 6 snare pins, but a pretty standard number, even though it's not as precisely known as it is here. And that maybe what's special about the synapse is that it has 12 snare pins rather than 6 to power the arrangement. It's turbocharged. And if you look at the structure that I've outlined for you, it's admittedly still a bit speculative, but in that structure, the peripheral snare pins are peripheral. They are radially further out, right? So if they are radially further out, they're exerting their forces in a different way. And that's what's encapsulated right here. 6 central snare pins, 6 peripheral ones, each exerting force. And this force is further out than the central. Now, we know that when a snare pin zippers, it no longer can exert force because the 4-helix bundle is now assembled. There's no more protein folding to be done. That's a basic concept. So we also see that the central snare pins are closer to the fusion point, the contact point, than the peripheral snare pins. So this is now prediction, but it stands to reason that the fusion pore will actually be opened by the central snare pins, not by the peripheral snare pins. So that when the fusion pore first opens, which is when the content is first released, that's the physiologically relevant moment for a synapse, the peripheral snare pins won't yet have zippered. So that means, if it's true, the peripheral snare pins are there for the purpose, for some purpose other than the mere opening of a fusion pore. Why would they be there? Well, we contend they are there to exert extra force. Force that can be used to open this fusion pore faster, to expand it faster, or for other purposes. Well, we've made some effort. Fred Poncet in our team and I have made some very simple physical calculations as to what the effect of these could be. And if you think about it physically, the peripheral snare pins are actually pulling down on this membrane. They are exerting force on the lipid bilayer that surrounds the water, which is essentially an incompressible fluid. It can change its shape, but not its volume. And we know the membrane tension in this vesicle, and we know the amount of force that a synaptic vesicle, that these pins can exert. So from that, we've calculated, and you can see the calculations if you wish, that the hydrostatic pressure inside this vesicle will become raised to about 3 times that of the atmosphere. And that raised pressure is going to cause a force vector on the fusion pore that, when it opens, is going to push the fusion pore open radially 5 or 10 times faster. It's going to cause the fusion pore to open in the first instance 100 times faster. And if this is correct, may even eject the fluid rather than simply allow it to diffuse out of the synaptic vesicle. So how can we test this? We can test this, if we could, by eliminating the peripheral snare pins. How can we eliminate the peripheral snare pins? Well, I think I've already told you how. We have the idea that VAMP forms a dimer, and the dimer binds to the synaptophysin. If we can eliminate the residues that stabilize the dimer, maybe then synaptophysin will continue to want to bind a dimer, but will only succeed in binding a monomer of VAMP, and therefore now have only 6 copies of VAMP bound to it. And in fact, that's exactly what happens. When we take the VAMP dimer mutant in the transmembrane domain, and we put it into our reconstituted system, now we get only 6 copies or 6 snare pins per vesicle. What happens when we test the kinetics of 6 snare pins per vesicle? This is the work of Atruli Chatterjee in the lab. Here are some traces of vesicles which have bound, and now they're released by calcium. You see they're synchronously released, and that's the wild type. This is the kinetics that you get for the wild type. Within the first time point, all the vesicles have released. They are synchronously released as advertised. Here's what happens if you have the dimerizing mutant. Then you see there can be quite a delay. This vesicle in particular I chose to show because it undergoes hemifusion and then complete fusion. This one was delayed 2 seconds rather than milliseconds, and it then released. But it's an extreme. More typically, the population looks like this. It's delayed to the point about half a second to a second. And so it's starting to look like all the other fusion events that we know about outside of the synapse. It occurs after a signal to fuse of about half a second to a second. And when it happens very quickly. This one was delayed 2 seconds, but when it happened very quickly. What happens to the release of content? I won't have time to show you the rationale of this experiment, but I will just show you some traces. This was done by Manindra Bera. And again, here we have 2 conditions. One condition where we have the 12 snare pins and the other where we have 6. And we're now looking at a vesicle that has bound. Its content is retained. The fusion pore opens when calcium is added. This is now when you have 12, the normal, the wild type. And then within the first time frame, the vesicle has released its content. These are 2 separate vesicles. Synchronous release. All within actually our first time point, which is 5 milliseconds. Now, this is a higher speed than the time resolution than the previous. Now, what happens when we have 6 snare pins? We see actually a rather different behavior. Now, the signal in the vesicle, which is retained locally, goes up, 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 and now goes down. What's going on? What's happening is the fusion pore is opening, closing, opening, closing, opening, closing in every case, and the amount of dye is going down, 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 but it's being open. It's being released with every fusion pore opening, but incompletely. The concentration of the dye lowers, therefore it starts to dequench. It reaches the point where it's maximally dequenched, and now the total signal starts going down. So that envelope represents an envelope of 20 to 100 milliseconds over which the vesicle is engaging in essentially kiss and run. It's not actually leaving, but it's opening, closing, opening, closing, opening, closing, controlled in this case by the number of snare pins. So the peripheral snare pin then is required, as you can see, in fact, is responsible for the rapid release that is observed in the synaptic system as it's reconstituted. When you take it away, you get kiss and run, which is very, very interesting because people have observed it, of course, in neuronal synapses, but not really been able to regulate it or understand the best structural basis of it. We have a possible explanation of that now. And I will skip that in the interest of time and simply point out that current focus in the lab is the now challenging. You can imagine that the structure, we've got a glimpse of it that exists under each vesicle. It's not just one structure, it's at least 3 structures that are involved in the assembly of the vesicle. In the readily released form, we can count from symmetry at least 108 polypeptides that form a structure that's 3 times the volume and twice the mass of a ribosome. And it may even be more at certain stages if we include other proteins that are known to come and go. So we're hoping over the next few years to solve these structures at high resolution by cryo-EM and flesh out the view that I've outlined for you, which has probably raised more questions than it has answered, but it at least allows us to frame hypotheses now with real precision about how how the synapse is able to achieve turbocharged release, and it introduces a whole series of stages of precise, potentially precise regulation, including the regulation of incomplete neurotransmitter release, that can probably be studied in ways that, by physiology, that will be informed and enlightened by structural biology and by cell-free biochemistry. So I'm sorry that the talk has gone on a bit longer than expected. If anybody wants to stay for questions, I certainly can stay for questions and would be happy to do so. And again, thank you to our host for enabling me the opportunity to share this data with you, almost all of which were very recently published in the matter of last few weeks or month or two, and some of which, a good deal, actually not yet published.